You realize how short you are when you get to the podium and you have to push it down so far. But yeah, okay, so we have a very interesting session coming up. I just first like to welcome everyone back from the snacks and the lovely chocolate milkshakes we had. So I'm Anja Bierke. I will be running this session today. I am our resident psychologist. So if you have problems, I really don't care. Okay, so we're going to kick off the session with Nicki Tiffin, who is, along with myself, the co-chair of the PHA4GE Ethics and Data Sharing Working Group. Lovely to have you, Nicki.
Great, thanks very much. It's such a pleasure to be here. And I'm just going to jump right in. We're talking about ethics and equity in microbial data sharing. I think there are a lot of principles that are wider, but I'll try and talk about some of the work we've been doing in the space and with PHA4GE and other partners, microbial partners. Just to say, yes, as Anja mentioned, I chair the PHA4GE Ethics and Data Sharing Working Group. So I just wanted to say that we work slightly differently to the other working groups. We're more responsive, so people contact us when there's a challenge that they're facing, and then we stand up a group, and we work from that with that group to address the issue that's been raised, try and build something to make it a bit easier, and then from there we close down that group and wait for the next thing to come in. So we're putting it out there. If there's something that you want us to look at, please do use the QR code. There's a form, and just let us know, and we'll see if we can help you out. And I'll put the QR code up again at the end. Yeah. And also, if you want to be part of the people we alert when there is something that we are standing up to address, then also sign up on that form to get on our working group mailing list. So ethical and equitable microbial data sharing, it's, these terms everyone's using all the time. Equity is like the new catchphrase. So I thought I would just talk to all these things in terms of what's the principle and what's the practice. So the principle for equitable and ethical data sharing is really making the best use of pathogen data to improve health for everyone. And we can add by everyone there as well. But in practice, what that means for us is implementing governance frameworks, and personally for me, I like building tools to make it a bit easier. I think we need all the principles, we need the guidance documents, we need the high-level documents, but actually as researchers, we want tools that just make it a bit simpler. This is kind of the governance infrastructure I normally work around, and I'm not going to get into it in a lot of detail, and mainly also because I can't read on my little slide guide here all the small text. But essentially, there are these different pillars of equity, ethics, legislation, and also procedural and structural controls, sustainability. These things all together make up what constitutes governance and a governance framework. But here I'm going to tell you more about the practical stuff and some of the things we've been doing in the working group, and then also a little bit more widely as well. So the principle, I'll tell you what the principle is that we're addressing, and then kind of what we've done to try and actualize it or operationalize it. This principle was brought to us in the working group about how we do benefit sharing. The principle is that we want to share the benefits of our work more widely with the general population, with researchers from different parts of the world. But in practice, what we did was we built a benefit sharing framework. It's like a two-dimensional matrix that aims to help researchers situate the kind of benefit sharing they can do, not only researchers, actually all stakeholders, and fit things together. And some of you have probably seen this. I think we may have presented it at the last meeting. It was our first product out of the working group. And this is, just a link. We ask in this framework, we try and make it simple to see if the research is contextually relevant, is it useful for the population it's supposed to serve, and then also about benefits not only to the participants, obviously, and the population where the work is being done, but also to the researchers, recognition of contributions. So it's a large matrix, and it should be easy to use once you get going. Then we also were asked, I come from a human genomics background, and we kind of know the baseline rules. We know the etiquette of working with human genomics data, but we were approached because it's not always so clear for microbial data, like what's the basis and what's the common standard of acceptable behavior. So I think at the last meeting, we actually put this out to one of the working group meetings to start helping us shape this PHA4GE Microbial Data Sharing Accord. And we published that shortly after the last meeting, I think. And what this is, it's a kind of set of the most basic common decency rules if you're working with microbial data. And we called it microbial data because something that's not a pathogen here might be a pathogen there, etc. Obviously, it's also, was coming out of the pathogen kind of community. And we set these kind of basic rules, and the idea would be that this is a starting point when you're negotiating your sharing terms and conditions, and you might modify it, but at least you could say, okay, I'm sharing these data according to the PHA4GE Data Sharing Accord, and everybody kind of knows what those points are. So we had 7 attributes, how you attribute the people who generated the data, who should overview your publications before you put them out there, and how you should, whether you can share the data onward or not, data anonymization, geospatial data, intellectual property, and opportunities for collaboration. And the idea would be that you could modify any of these. It's not a set of rules that has to be followed. It's a starting place to negotiate next steps. At least you all start from the same place. Then we were also approached about wastewater surveillance. I changed this slide a few times because I think it was actually more like ethics for wastewater surveillance is utterly incomprehensible. But I don't want to be too negative putting it up here, so I just said difficult. So what we did was we built an app that, it's like an alerting app. And the way it works is you can put in, here it is here, you can put in exactly which kind of data you're working with in the wastewater that you've generated the data from. And according to what you select, are you working with DNA, are you working with RNA, maybe you're looking at metabolites, is it human, is it animal? You can select these from the drop-downs, and as you select, the bit that is below will populate with specific advice on what to think about in ethical terms as you work with the data type. It's not a solution that's going to give you all the answers. It's going to tell you what to think about. If you're looking at plant DNA, or you have plant DNA in your sequence that you've generated, it's going to remind you about the Nagoya Protocol, for example. And if there's human DNA in there, it's going to remind you about the basic ethics principles. So it looks at legislation that might be relevant, and ethics issues as well, and then equity and kind of justice issues. And there is a little note that if you're working with the whole sample, everything applies because everything is still in there. So yeah, just to put in that little disclaimer. Then, we have some other projects on the go. So this one is about how we can communicate data sharing requirements to end users. This is, for me, is a very exciting project, and it's coming to fruition. We are building a data sharing license. It's pretty close to completion. It'll be live in December at the latest this year. So just to before going a little bit more into the data sharing license, is just to mention this idea that, and it's come up quite often in the discussions, that we are actually moving away from this idea that data sharing means I have data and I give them to you, and that's the end of the story. And it's a lot more nuanced now, and there's a lot more to communicate when you want to share data. Maybe you have data that you find for people to put in their dashboards, but you don't actually, as an aggregate count, but you don't actually want them to go and do some kind of academics research and publish, etc. There's so many more layers of what can and can't be done. We tried to sort of put a name on the different types of sharing and put it in one place, and we published a paper actually talking about what is federated analysis, what is a TRE, a trusted research environment, what do we mean when we talk about collaborative sharing as opposed to direct sharing, which is what we all intuitively think of. So the data sharing license is designed to kind of capture all these nuances and put them into one place, into one document that clearly communicates exactly what is or isn't permitted with the data set. So who is familiar with Creative Commons? I'd love to actually see a show of hands. Great, actually, a lot of people, good. So this is, I even put a little Creative Commons in the top right there for my material, just as a sort of memory jog for everyone. So the data sharing license it's inspired by Creative Commons in that it's like a Creative Commons type license, but for data sets. So we have a whole lot of different points where we ask prompt for information about the sharing conditions, and then depending on what you select, a particular license is generated. So we're going to launch this as it'll be a website similar to the Creative Commons license chooser, where you can go and answer a series of questions and click on your yeses and nos, and at the end, as you finish building the license, it will pop out a series of different outputs. So there will be a PDF document that explains exactly what each element of the license means. We'll have a line of JSON, so all of you who want to just suck it straight into your databases, that will be ready. We'll also have a text-based license variable if you're working in flat files. And then, of course, we'll have a very beautiful icon for you if you want to actually have something pretty. So we're working with our dev team here, Method Lab, they're fantastic, and also Homonym, who are currently just cleaning up these logos. This is my PowerPoint version of logos, and they're turning them into something beautiful and functional. So we're very excited to launch that. Yeah. And then this is also just a project of mine that recently launched, and it's hopefully useful to pretty much everyone in this room. The principle is that Africans need a fairer deal when they share their data and biospecimens, and I won't get into a rant on the history of why this needs to be so, but just believe me, it does. So we've been working on building a platform, also actually with Method Lab and Homonym, our partners, and we've built what we call the African Data and Biospecimen Exchange. And what this is, oh, and just to say it's live. Yeah, it went live maybe 2 months ago. We did a kind of soft launch. So currently it doesn't have huge amounts of data in there, but people are using it, and we'll be doing a rollout across the continent as well. So over time it will gain more data, obviously, and more resources. And yeah, the way it works, African Data and Biospecimen Exchange, it's ADBEX for short. It's not I'll tell you what it isn't, because people get a bit confused. So it's not a data repository or a biospecimen repository. Nobody sends us anything. In fact, we really don't want your precious resources. I really wouldn't like that kind of responsibility. And it's also not a replacement for anything that's out there already. So some people have stuff in open databases, some people have stuff in closed databases, some have stuff in something that's halfway. This is another layer that can sit above there. It's not intended to take over a role from anything else. And in fact, early in the new year, we will be running an API workshop for people who have large repositories who would like to use this to automate how you can advertise what you have through ADBEX. It's also, yeah, it's about bringing together or connecting people sitting in Africa who have data and biospecimens and people out there who would like to share them. And so if you have African resources, I'll call them resources because saying data and biospecimens gets a bit tiring for everyone, me included. So if you have resources that you're willing to share, you can list them on the platform. And then everyone in the world can have a look if they're looking for resources, and they might find something through the search that they really like the look of. There's a messaging function in the platform, so if you do like something, you can message the custodian or the responsible person for that resource, hey, I like that data set, are you open for sharing? If you can chat a bit, come to an agreement, at that point you can also bring in your PI or your legal team or your TTO to the conversation. Talk your way to wherever you're happy with the arrangement. It gives the Africans who are providing the resources an opportunity to say, well, I really like that, but could my student come and learn the technique while you're doing the analysis, or let's collaborate, or it's only available for federated work. Once you've agreed all of that, there's kind of I always call it a wizard, but it kind of gives away my age, but a builder, and it will take you stepwise through building your data sharing agreement or your biospecimen sharing agreement. And that provides like an MOU. It's not a legally binding document, but it will have all the necessary information, all the clauses for your data sharing agreement, and you can give that to your legal office as you need to build the legal document if you're doing something cross-border or whatever the requirements are. Yeah, so it's an idea of just bringing a more level playing field to the sharing arrangements. I encourage all the Africans in the room to please go and list your resources, and I encourage everyone else to go and search them, and let's get some really equitable data sharing going. It looks kind of like this, or it does look like this. That's a landing page, and as you can see, there are like two personas. They're the people who are providing the resources and listing them, the people who are searching the resources, and it leads to discussion, building MOUs, maybe working together, maybe just handing over the data or biospecimens, depending on each choice amongst the partners. So yeah, I think that's pretty much it from me. Just reminding you again, if there's something you want me to stand up a group to look at, to address in the ethics equity space, we'll be very happy to certainly see if we can help stand up a group to tackle it. So do go on to that form. It's a Google form. It's also on page 89 of your program if you don't catch it here, or you think of something at 4 a.m. tomorrow morning and you want to send us a message, we'll leave it open, so keep on sending those in. Or sign up if you want to be on the group that responds as well. So yeah, just to say thanks, and really to recognize all the partners. So thanks to the Gates Foundation who funded the ADBEX platform and the data sharing license. And then to say really my very valued partnership. I'm currently in Berlin for obviously I'm here, but in Berlin for 6 months on the WHO Berlin Hub and Charité University inaugural Public Health Intelligence Fellowship. So I'm one of the fellows there for 6 months, and this is where we're developing the data sharing license as my project under that fellowship program. So yeah, and of course, thanks to the team, thanks to PHA4GE, and thanks to all of you. Thanks.
Thank you, Nicki. We really appreciate the work you and your teams are doing towards developing ethical and equitable pathogen data sharing through tools, frameworks, and guidelines. For our next speaker, we have Natalie Groves. I actually asked her, and I did it wrong in any case. Groves. From the UK Health Security Agency. Thank you, Natalie.
Hello, everyone. Thank you for inviting me to speak today. I'm going to talk to you about the our genomic-based risk assessment of avian influenza and how we use that to respond to a recent human case in the UK. So just a bit of background on avian influenza for those of you that kind of aren't familiar. Avian flu is either categorized as high or low pathogenicity, and that's defined based on its ability to cause severe disease in chickens. And it's primarily determined by a kind of cleavage motif in the HA segment and not really by much else. As everyone will know, the influenza is kind of subtyped based on HA and NA, but H5 is also classified into clades using the hemagglutinin phylogeny, and the clade 2344B is currently responsible for kind of a global panzootic. So this clade emerged in 2020 to 2021, and it was quite different to what we'd seen before in avian influenza because it wasn't really managed by normal animal health practices. So culling on farms or housing orders didn't really seem to kind of reduce the burden. And we also saw a kind of expanded host range. We were seeing lots more shorebirds, migratory birds, and this contributed to a global spread. The clade also caused quite a lot of mammalian infections, and some of those were quite big outbreaks. So you might be familiar with some of the fur farm outbreaks in Europe, or the pinniped outbreaks in South America, and most recently the cattle dairy farm outbreaks in the US. Alongside that, there were quite a lot of human infections. So in the tree on this slide, all the human infections are kind of in red, which hopefully you can still see, but they're kind of across the tree, and there were quite a lot of them within this kind of 2344B clade for people who were kind of working on farms or were in environments that were contaminated. So how do we actually assess the risk to a human from a sequence that came from a bird? It's unfortunately not as simple as just looking for a cleavage site in HA, but we're primarily looking for 4 viral characteristics. The first one is polymerase adaptation, and that is kind of mutations in any of the 3 segments that are involved in the polymerase complex, and the mutations that we see in these segments will increase replication in mammalian cells. We also look for resistance to host restriction factors, and these are kind of less likely to immediately appear in a mammalian host and are sort of more genotype to genotype specific, but are based on caused by mutations in the NP segment, and will basically just mean that your kind of innate immune response is less effective if you do come into contact with this virus. And the last 2 are the most important, but also the most complex, and they are based on the HA segment and the kind of host specificity of that. So we look for changes in the kind of receptor binding specificity across HA, and that is highly host-specific, but also tissue-specific. So you will see different tissues that are infected in different people based on this receptor specificity. So in birds, you see what we call alpha-2,3 linked sialic acid receptors, but in humans, we have what we call alpha-2,6 linked receptors. And for effective infection and host cell entry, but also between host transmission, you need to have these kind of receptor-specific changes in HA. The other thing that HA needs to be able to do is persist in different environments. So the core temperature of a duck is lower than the core temperature of a human. So the HA kind of mutates to survive at different temperatures and kind of persist in different parts of those hosts. In birds, it tends to be more of a gastric infection, and so their transmission is kind of fecal-oral route, but for humans, it's obviously a respiratory infection, and we see airborne transmission. But you don't really get that airborne transmission for avian influenza because it doesn't have these HA changes. So into the meat of the actual kind of assessment that we did within the UK. So our animal health colleagues share sequence data with us routinely for anything that they generate from their own surveillance. And we then assess that based on those kind of 4 principles that I just talked about to evaluate the current risk to human health. So looking at the data that they provided us, we were able to say that in January 2025, DI.2 was the predominant genotype that we were observing in UK birds. From the publicly available global data, we were also able to say that this genotype had been predominant across Europe the previous avian influenza season. And that meant that we could kind of assess how many mammalian outbreaks we'd seen, how many human infections we'd seen, how severe all of that seemed to be. we could show that there were no core mutations present in this genotype that would affect any of those 4 characteristics that I was just talking about, and the HA was consistent with avian receptors. We had kind of early in January, we'd had a cluster of sequences from an infected premises in the UK that did have a polymerase mutation that would be associated with mammalian adaptation, but that premises was managed by our animal health colleagues and was considered to be kind of a closed outbreak. We weren't concerned that mutation was spreading across the UK birds. So overall, we assessed the season as low risk. And that meant that we were only really managing high-risk exposures with antivirals and close contact tracing. So somebody that was exposed to infected animals with no PPE or a breach in PPE, or was exposed to a confirmed human case. And if you can manage a situation in that way, it obviously really reduces the burden on your public health teams because you've got fewer people to follow up, you've got fewer antivirals to try and kind of spread everywhere, and you've got fewer people that have to go into a high-consequence infectious disease unit in a hospital, which have very limited beds. So obviously we were notified about our UK case on a Friday afternoon, because why would it be any other time? But we were able to produce sequence data, not just from the human case, but also from the poultry on the farm within 72 hours of the human sample collection. And this was really valuable for us because we were able to show that the sequence from the human was exactly the same as what was seen in the birds. We weren't seeing any adaptations that were host-specific, and it was consistent with that DI.2 genotype that we'd previously assessed. So that meant that we could just manage this single exposed person, we managed their close contacts, but we didn't have to do any kind of immediate vaccination, any extended contact tracing, and once that person kind of recovered and had isolated, we were really only managing people on the farm that were exposed to the birds. We weren't concerned that there were kind of onwards transmission chains that we weren't going to be able to control. So you can see in the tree as well, the sequences that are labeled, they're the sequences from the chicken and the human in this situation. And they're kind of different from the rest of the UK cases, which are in purple, but still kind of clustered roughly with them. And that's really just because the surveillance in the birds is quite sporadic. We don't have a lot of surveillance across wild birds, because funnily enough, they don't submit to sampling just walking into a public health lab. And we only really see sequences from infected premises, and the kind of aim is that we would get at least one sequence from each premises, but as many as possible. But it does mean that we have to kind of infer things, and there can be quite a lot of mutations between what you've kind of assessed and analysed and what you actually see in your sort of human case. But luckily in this case, there weren't too many mutations for us to look at, and we could do that rapidly. So we had sequencing within 72 hours, and then within kind of the next day, we had all of our reports shared, and we were happy that the public health response had been proportionate and we didn't need to upgrade any of those response efforts. So just to kind of finish with some sort of summary points, the kind of proactive assessment of this sequence data from the animals was really helpful in our rapid response to a human case. We already knew a lot about what was circulating and what that would mean in terms of our response. And that really depended on us identifying these kind of genomic features that would inform the risk to human health, and a lot of that comes from our academic collaborators. But collaboration between our animal and human health agencies is obviously super important in this. If they hadn't sent us any data, we wouldn't have been able to do those risk assessments. And similarly, kind of the global contribution to sharing sequence data, we can't tell anything about what's going on in the animals if no one shares anything with us. But the surveillance of those animals is really important. So that a few times kind of throughout the conference, we've talked about funding and prioritization of that. And I think it's really important that we don't just focus on sequencing the pathogens from the humans, but we need to think about this in a One Health way and where those infections are actually going to come from. Because if you understand that reservoir, then you understand the risk to people much better. And also, just because metadata is everything, this is, I guess, just a plea for like sufficient and consistent metadata, because if you don't know what host your sequences come from, what country it was detected in, when it was collected, all of this is entirely pointless. So yeah, please, please include your metadata. But that's all from me. Thanks.
Thank you, Natalie, for showing how a One Health genomic surveillance approach can strengthen genomic risk assessment and response coordination. I think anyone within the field of One Health has realized how important it is that we look at all the different sources. So our next speaker is Idrissa Dieng. I'd like to invite him here to the podium. He is from the Institut Pasteur de Dakar.
Thank you so much for the introduction. For this talk, I would like to share with you some data that we have about the genomic surveillance of dengue in Senegal. And here, this is just the layout of our talk, what we will talk about. A general overview of the emerging disease, a focus on arboviruses, and then the real-time tracking of dengue variant that we are working on in Senegal, and also the genomic characterization of the strain that we have inside the country, and finally, what are the impact of this genetic diversity on diagnostic and countermeasures. And then, as summarized by this picture, it just tells us that emerging infectious diseases are bad, and they are associated to high burden on public health. And among the viruses that are associated to this, we have the arboviruses, that are a group of viruses that are unique on the transmission model because they are transmitted to one people to another using the vector that can be mosquitoes and ticks. And here we will talk about the viruses that are mainly transmitted by mosquito vectors. And then we will make a focus on the dengue viruses that becoming to be a major public health threat worldwide. And we starting to see some cases of the virus in some temperate regions where the viruses was not known to circulating before. And then for these viruses, we have two circulation cycles, two epidemiological cycles. We have what is called the urban cycle, for which we have the circulation of the viruses between the human and peridomestic mosquitoes of the genus Aedes albopictus or Aedes aegypti, what is the main viruses, the main vector of the transmission of the viruses. And also beside, we have what is called the salvific cycle, where we have the circulation of the viruses between the human and also the non-human primate and some arboreal mosquitoes like Aedes aegypti, Tylori, or Frucifer. And clinically, the infection of arboviruses are slightly the same. You have the febrile illness, and for some cases, they can evolve to cause some hemorrhagic fever cases like what we can see on dengue viruses or Rift Valley fevers and others. And the map summarizes what is the global repartition of dengue viruses. You have dengue viruses circulating all around the world, mainly in the tropical and subtropical areas. And then, according to the distribution of these viruses, it is a time to have some studies that highlight what is the way that we can improve the diagnostic of the viruses, how we can improve the genetic surveillance of the viruses, and how we can manage to reduce as soon as possible the virus's burden. And then this slide also summarizes what are the parameters that are linked or shaping the emergence of the arboviruses. We have the climate change. That is a big problem because due to the climate change, we have the circulation of the viruses in different areas where the vectors are not distributed. And then the situation was observed in France, where we found that in 20 years, they have a wide spread of the albopictus vector that are transmitted the virus. And also one other parameter, it is the urbanization that can cause the habitat disruption. This means people are more and more living near to the place of emergence of most of those viruses. And also one parameter, this is migration. With migration, for example, one virus can emerge in Southeast Asia, and the day after, these viruses can spread worldwide and then cause a huge outbreak. In Senegal, we have a system that is called the 4S network. This is the Sentinel Syndromic Surveillance Network. That is, the idea behind it is we have a Sentinel site in each of the 14 regions of the countries. And the aim of this Sentinel site, it is to capture any habitual health event. For example, in one place, if we see that have an increase of the number of febrile cases, it is the signal to take the samples and to ship it in the reference lab for arboviruses and hemorrhagic fever virus that is located at the Institut Pasteur of Dakar. And there, once we receive the sample, we are running a set of assay and testing for looking for routinely up to 7 arboviruses of medical interest, including dengue, Zika, chikungunya, Rift Valley fever, Crimea Congo, yellow fever. And then we are sending the result that we got with the Ministry of Health in order to organize the appropriate response in order to reduce as soon as possible the burden of the viruses. And then recently, we are adding a layer of the genomic surveillance of this one. This started in 2017, because before we are just limited on the detection of the viruses, let's say this is positive or negative for one viruses, and we add a layer of the genomic surveillance in order to track in real time what is the variant of the virus that are circulating around the country. And then for this example of dengue, we are using some tools that are available in open source. And also for some part of our study, we designed some tools that help us to answer specific questions for which the tools that are available are not able to answer. That's why we are designing a tool that is called Omicron, oligo mismatch counter, that allow us, for example, if we have a variant of the virus, and near to this variant we have, for example, the primer scheme that allow to sequence the virus, we can know if this primer scheme are enough closest to the variant of the virus to sequence this virus instead of going to the wet lab part, running the sequencing, and so that we have a dropout in some region of interest. And also we made one tool that allow us to generate primers in case that we found that one RT-qPCR assay is not longer taking a variant of the virus. And also we are using tools that are available like NextRAN for phylogeography and others. And this slide summarizes the data that we have around this the surveillance of dengue in Senegal. As you see, we started in 2015, and through years we have seen an increase of the number of dengue cases that was detected around the country. Just as an example, the last year we have up to 900 infection of dengue cases detected by PCR around the country. This means that in Senegal, dengue is becoming a major public health threat. And then, according to the data that we are generating on the genomic surveillance, we are tracking in real time what is the variant of the virus that is circulating. What you see on this slide, it is through time we have the change of the dominant serotype that are circulating around the countries. We have increased number of dengue trees that replace all the other serotypes through years. And then recently, the 2 serotypes that are circulating around the countries are mainly dengue viruses 1 and dengue viruses 3. And we saw also a phenomenon of serotype replacement, and through time, maybe this is due to the fact that the variant of the viruses that are circulating are more adapted to vector or are more transmissible. And some previous work has shown that the genotype of the dengue virus 3 is more transmissible. And also at the genotype level, we found that as far we have the circulation of 3 serotypes of a virus, we have a lower genotype diversity. This means for each of the serotype, we have just one only genotype that are circulating, except for dengue viruses 2, for which we have 2 genotypes. And also at the minor and the major lineage, there are the same that are circulating, but they are belonging in different clades. This may be sufficient to sustain a transmissibility of the virus and a permanent epidemic through years. And then this global map summarizes where we found in the world this genotype of the viruses that we have detected in Senegal. And then I will dive also to another part of this talk, and I will start it with the fact of Louis Pasteur that said that change favors the mind that are prepared. For example, in order to see one phenomenon that are strange and others, and found that this phenomenon is related to this or this, you need to have a prepared mind. And also another aspect, this is the pictures that are summarized the history of Achille. For Achille, their mother wanted to protect him, and for this one, he dived Achille on the river of Styx. And then by holding it on the water, he held it on the heel. This means that all the part of his bodies that are touching the water were protected except the heel. And for the diagnostic of the arboviruses, they are Achille heel that we will talk about in detail soon. And the history that we have here, it is Senegal in the southern part of the country. In 2021, we have a dengue cases that we detected around the countries. The patient have the same clinical pictures of all the dengue cases that we faced before. For this patient, we collected the blood sample and shipped them to the IPD reference lab. We run the PCR for these samples, and then we wanted to move forward to do the molecular serotyping using RT-qPCR. And in our surprise, the sample that was positive with a CT value lower than 26, we was not unable to assign the serotype to this variant of the viruses. That was something that is strange for us, because it makes no sense that we have a higher CT value, and this particular sample was not able to be assigned to a serotype. And then we tried to find what is the logic behind. And then for this one, we are using a sample workflow, and the workflow was for arboviruses and principally the flavivirus. There are one gene that are conserved among all of them. That is called the NS5 gene, that is corresponding to the polymerase. We have a set of primers that allow to amplify all the viruses of this genus. We use this set of primers, amplify the lengths of 1 kb, and after we run the sequencing using nanopore, and after the blast result, the blast shown that the dengue viruses that we have in these human cases, it is the viruses that is belonging to the salvatic genotype of the virus that is known to circulate to non-human primate and arboreal mosquitoes. And then from this partial sequence that we got, we make the blast, and then from the blast, we just pick the 15 closest hit for which we have a complete genome, and from this, we design a new set of primer scam in order to amplify and get the full genome sequence of the salvatic strain that we detected previously. And then using our tool Omicron, we try to assess if the scams that are available for dengue viruses 2 are still able, and what are the level of similarity with the salvatic strain. In the box below, you shown that for all the salvatic strain that we analyzed, they have a higher number of mismatches against this strain. And this explains why when we try to sequence this virus using this particular stem, we have a lot of dropout to sequence them. And then in response to this, we designed the set of the new primer scam, and in this figure, we compare what is the evolution of the coverage that we got using different scam. We used the one published in 2019 in Angola in response to the dengue outbreak, and also we used the one that is published by the Yale University that is called the pan-dengue sequencing. And both of them are showing that we have a set of dropout in different parts of the genomic region. The only scam that provides a full and a consistent coverage across the genome, it is the new one that we designed for salvatic dengue virus. And then also, we wanted to explore in detail what was the reason why we was not able to assign a serotype to this particular salvatic strain. And by aligning back the primers and probe that we have in the CDC assay, we shown that we have a higher number of mismatches on the primer binding site, particularly on the probe for which we have 5 mismatches, and they are all located in the critical region of the viruses. And then in response to this one, we decide to set up a new RT-qPCR primers, and then we spike it on the multiplex assay in order to be able, in one reaction, to detect the urban dengue viruses 1, the urban dengue viruses 2, the urban dengue viruses 3, and the salvatic dengue viruses 2. In this particular assay, we was not able to put dengue viruses 4 because we was limited in terms of dye that we can use in the reaction. And what we see, both in singleplex and in multiplex, the new assay that we designed is able to detect and assign the salvatic virus lineage, and this can be a useful tool that we can use in the One Health approach in order to see what is the prevalence and the level of circulation of this particular variant of the viruses among the mosquitoes and among also the non-human primate, and also we can use it for human. And this particular work was use it in these 2 publications. The one just to show that CDC Tiroplex assay can miss some variant of the dengue virus that are circulated in Senegal and in West Africa, and also the new set of primer and the RT-qPCR that we designed in order to be able to detect in one round reaction the variant of the viruses that we are dealing with. And also we're trying to using this genome to do some in silico approach by comparing the salvatic strain and the urban, and we saw that in terms of codon usage, they are using different set of codon in order to amplify and translate efficiently that amino acid. This means that these 2 viruses are mainly different way to replicate on to propagate in cell culture. And then using also what is called the codon adaptation index. This is, for example, as you may know, for the viruses, the genetic code for the organism, the genetic code is degenerated. For sequencing the protein, they have the possibility to choose one of the combination of codon to set it. And as far as the codon that is used by the host to translate to some housekeeping gene are matching the one that is used for the virus, we assess that the viruses have a high capability to replicate. And what we see here, it is none of the contemporary salvatic strain that we detected in Senegal have a gain of adaptation in human or in mosquitoes. And then also we compared the adaptation between human and mosquito. We saw that we have a higher correlation. We have a p-value, significant p-value as a low correlation, meaning that this is not only the genetic factor that are contributed to the emergence of the salviatic viruses. And then we perform some phylogeographic analysis in order to see what is the time of the TMRC of the cluster of the strain. That we shown that the TMRC is dated back to 2004 and highlighting that maybe we have a critical circulation of these particular viruses in the Senegal, and then we have to move toward in order to see what is the prevalence of the viruses in the on health environment. And also we were surprised days ago to find that the same salviatic strain that we seen in Senegal re-emerged in Malaysia, in Southeast Asia, in Malaysia. And this particular virus is belonging to a different clade that is different from the one on Senegal. Just to let you know that beside the urban dengue viruses, we are facing more and more the re-emergence of the salviatic strain that can add a layer of complexity in our understanding and the countermeasure that we are using for the viruses. And finally, we wanted to know what is the level of the proximity of the viruses, the main variant of the dengue virus history that we have in Senegal against the vaccine for dengue. And we just compare in silico the viruses amino acid that we have against the two main vaccines that are proposed for dengue. And we found that for the immunogenicration of the vaccine, we have 22 and 23 amino acid change. This means that before any policy of vaccination, we need to take in account this fact that maybe the vaccine may not be effective to control the dengue viruses in Africa. And then we split in order to see in which domain we have the most of the amino acid change. And it showed that most of the amino acid change are located in the ED3 that is known to play a crucial role on the virus's neutralization. And also we map some change that we got on the out of the map or the B cell that we found for dengue, and we showed that the viruses that are circulating in Senegal are exhibited 2 amino acid change on the map. This is what I would like to share with you. If you want to learn more about our work, you can just scan the QR code. Thank you so much.
Thank you, Idrissa, for showing how nanopore sequencing was used when serotyping failed for sentinel syndromic surveillance of the dengue virus. It was very interesting to see how you repurposed. For our final talk in the session, before we get to Q&A, I ask that Celeste Donato join me. She is from the Peter Doherty Institute for Infection and Immunity.
So thank you very much. So today I'm going to be presenting on a One Health program that we've been developing as part of the Oz Pathogen program in Australia. So the Oz Pathogen program is the largest pathogen genomic translational research program in Australia that brings together experts and academics from all states and territories across the country and has fantastic representation from public health laboratories and departments of health. The overarching aim of the program is to optimise national genomics-based surveillance of infectious diseases. And this program is underpinned by the AusTracker platform that you heard my colleague Claire present on in the prior session. We have a number of different pathogens within this program, and the E. coli One Health project is one of them. So the project had 3 separate components. We had a retrospective project where we analysed all of the published and in-house E. coli collections across Australia over the broadest time period that we could account for. And we did this to get a bit of a picture of the diversity of E. coli in Australia, because we have very diverse ecological niches and lots of different contexts that would impact diversity and transmission events. We also used it as an opportunity to start assessing different tools and methodologies for a One Health analysis context, because of a lot of the tools that we use in public health contexts aren't necessarily directly applicable to One Health analyses. And whilst we were analysing this retrospective data, we started planning our prospective study, which was the largest component of this project. It was a 12-month snapshot that we conducted just within the state of Victoria, because Australia is a massive country and the state of Victoria alone is larger than most European countries. We sort of had to restrict ourselves geographically initially to start planning this program. And we did this to assess different sequencing strategies across different sample types and really think about the feasibility and utility of routine surveillance for E. coli as a One Health scenario. And the third major component of this program was stakeholder engagement, where we had a lot of focus on workshops and training with all of our stakeholders and our key industry partners. And this was very much led by the industry and the different sectors to facilitate these workshops, because we needed to know what they wanted out of this type of surveillance. So when we came to design the snapshot, we really looked at that retrospective study. We looked at the sectors that had been poorly covered or not included at all to help design sectors to focus on. Sample types that had been missing historically from One Health collections were also a primary focus, and underrepresented groups. Again, we really looked at engagement with stakeholders to ensure that we were meeting the needs, and also that our reporting would be tailored in a format that would be useful to stakeholders and that they could use as actionable outputs. So first off, we designed the strategy to think about all the different sectors that we were going to sample across this 12-month period, how often we would go back to each different sampling opportunity, how many samples we would collect at each point in time. We had a robust laboratory methodology for all of the different sampling types that we collected, and we were doing both Illumina sequencing to begin with, and we will go back and do some ONT sequencing as well. And we've also developed a lot of different pipelines and tools to analyse the data, but also to embed some of the unique visualisation requirements within the AusTracker platform. And this entire project had a governance of 3 structures. We had the AusPathogen executive, which oversaw the entire AusPathogen program, but then we also had a One Health exec and a One Health working group. And the One Health working group had over 40 members from all different sectors and academic backgrounds. And every single decision that we made, from data governance, ethics, metadata variables, sampling strategies, sampling plans, stakeholder engagement, every single decision was a decision made by committee. We wanted as much perspective as we could get on every single decision that we made. So these tiers of the executives and the working group were really critical to make sure that we were thinking about every question the right way and that we were engaging with every stakeholder in the most appropriate way for their needs, but also for their concerns. We spent a huge amount of time mapping all of the stakeholders that we thought that we were going to be able to engage with for this project, and identify the key people within those different sectors and start thinking about the tailored requirements for sampling of those sectors in terms of data arrangements, MTAs, ethics, and all of those kinds of considerations. And it took over a year and a half to map out all of these different sectors and players and start getting the agreements in place to actually be able to get the samples into the lab. Some of these different stakeholders, it was an absolute dream. Like, we literally, buckets and buckets of samples were being provided. We couldn't keep up. Others were a little bit difficult. Some very terse conversations, a few people hanging up on me in the middle of phone calls. And it was really tricky. We didn't really know what we were doing, but we kept going back to the working group and kept saying, okay, we're running into problems here. We're not getting some great engagement there. What should we be doing? What are we doing wrong? Is it our communication strategy? Is it our approach? What's the threat? What are they perceiving? And the more that we sat down and talked one-on-one with people, we found they were afraid. They didn't understand what we were doing. there's a really bizarre perception that we hadn't thought about, was that DNA and genomics equals blame to a lot of lay people outside of what we do. And they were thinking, you're going to tell me that I've done something to someone else or to some other ecosystem, and you're going to blame me what's happened. And you've got the evidence, you've got DNA, I've seen CSI, I know that you're going to tell me that I've done this. And then we had, one industry person say to me, you're just going to tell me that I've killed a bunch of koalas. And I'm like, that's not what we're trying to do, but I understand your fear. So once we understood where they were coming from, we could completely change some of the language that we were using when we spoke to these stakeholders and tailor the communications to the level that met their needs and met their concerns. And that helped to some degree. Some sectors we still had a lot of trouble with, but at least, we made some progress. So we sort of started understanding some of the unique barriers and considerations to One Health in the Australian context. But I think this is applicable to most contexts when you're doing One Health. The lack of awareness and understanding around genomics, One Health principles and microbiology are really, really hard for some sectors to understand. There were pathogen-specific barriers. There are certain pathogens that are notifiable in livestock and agriculture, and even though we said we were only looking for E. coli, there was the chance that we could come across other things, or we were asking to create a biobank and potentially come back and sample these, use these samples for other purposes in the future. And so making it very clear what we could and could not do with those samples was critical. And we found that for certain industries, pathogen-specific agreements were the only thing that we could do to alleviate some of their concerns. And it's completely valid that they have these concerns. Fear of community perceptions was a massive thing when we were sampling public areas, swimming pools, parks, community farms, community gardens. They did not want people walking around with falcon tubes, with pipettes, looking like scientists, because again, it was this fear that people would think there was an outbreak. I think the pandemic is still very fresh in people's minds, and they still think that there could be lockdowns, there could be implications for the type of research that we were doing. We also had a lot of problems with anonymity. Groups happy to provide samples as long as it could never be publicly traced back to them. And that comes into some difficulties when it comes to analysing these samples and putting them in context of the rest of the collection. And we've tried where possible to mitigate and find a middle ground to these requests. Our communication strategy evolved over the project, but it was really clear that plain language summaries were critical and having really clear infographics. And usually, as funny and lighthearted and quirky as possible was the way to go. It took an incredible amount of time and resources to build those relationships and build that trust, and it wasn't something that we took lightly. We were really trying to understand what stakeholders wanted, and quite often they didn't know because they didn't understand the full potential of what we were trying to do. It was also really difficult in some circumstances to understand all the legislative and organisational barriers to participation. There'd be certain people that were really keen and they'd work their way up the chain and just hear a hard no, we don't do that. And then obviously all the permits, ethics, MTAs were, their own big set of challenges. And we also really thought a lot about how there was a lot of unmet need for sector-specific training, workshops, engagement on genomics. Even though we'd thought about it from the beginning, we'd really underestimated this need. So it turns out, we had a lot of conversations about what stakeholders in different sectors want. There's a bit of a difficulty in balancing their expectations around what's feasible, around turnaround times for analyses, around tailored feedback to each sector. The realistic, desires that they had were around data privacy and data agreements, some degree of tailored reporting within their sector that's specific to their data, actionable results, and upskilling of their workforce. Some of the unrealistic expectations that we quite often met were point source attribution for outbreaks on farms. This came back at us time and time and again, farms wanting this from us. And we just have to keep reminding them this was not the forum for that, and we weren't in a position to be able to do that for them, but we were able to talk to them about some of the tools and things that existed that could help them with those scenarios. We also had some issues around, certain sectors wanting access to all the data in the collection whilst keeping theirs anonymous. And, having some negotiations about whether that's reasonable and what we could do to facilitate a middle ground again. And also, we've also thought a lot about the need for further engagement and consultation with Indigenous communities across Australia and how we use this data in a respectful way for their communities. So in the end, we did a 12-month snapshot and we got an incredible diversity of samples within this study across every sector that we'd hoped to engage with. And we're really, really proud of this, and we're really, especially of our wildlife collection, because in Australia that's something that we're very passionate about, is our native ecosystems. So we collected over 1,600 samples for the biobank, and our strategy was to sample them for E. coli and store up to 20 isolates per sample. We've sequenced over 2,500 isolates to date, and depending on funding, we would have the potential to go back and sequence more. For most samples, we sequenced 1 colony per sample, but we sequenced all carbapenem-resistant and all SBL isolates. For human commensals, because of inherent diversity, we sequenced up to 10 colonies, and the same for environmental samples. We're only very early on in the analysis of this data, but we've got over 300 ST types, and we'll hopefully be producing some outputs really soon describing this cohort. The biggest challenge we had was metadata. We'd thought about this so much from the outset of this study, but even with all of the considerations that we had and the existing structure we had for the AusPathogen platform around metadata, we still ran into so many complications, and it's been amazing over the last couple of days to hear all the different approaches for metadata that we can now think about how we analyse some of our study in that context, but also how we might improve on our approaches in the future. Because, we had metadata that we wanted but couldn't have, metadata that we have but we have no clue how to use. Like, we had vets fill out pages and pages of history of some of these animals, and it's really valuable because some of these animals were, sick or had different interactions that could be relevant, but we don't yet know how to integrate all of that data together. And there's also some considerations of how much metadata we can realistically put into AusTracker while maintaining it as a reasonable platform that it's all usable in an intuitive way. And so we're playing around with lots of different approaches for that at the moment. So the next steps is, we're really focusing on integrating the genomic data and the metadata within the AusTracker platform and focusing on some of the unique visualization tools that are required for One Health data. We're going back now that we've generated some of the genomic data and really tailoring some of the teaching and training with specific stakeholders. And we're also generating case reports back to each individual sample provider. So over 80 different sample reports are going out with a pretty, low-level description of the diversity of their samples and a bit of information about what we've done so that the stakeholders still feel engaged in the process while they wait for the overall analysis to come, because that's going to take time, but we don't want them to feel like we've just taken their samples and nothing's happening and they're not getting any information. We're looking at scoping some of the national One Health genomic priorities across Australia, and we're looking to repeat the study in 3 years' time. So it just leaves me to thank the sample providers, all the members of the One Health AusPathogen program and the One Health working group, the members of the One Health exec, and the postdocs and lab team that were really critical in processing all of these samples across the project, and the AusPathogen partners as well. Thank you.
Thank you, Celeste, for closing our talks. I'll invite our speakers to come back to the stage. I know we're massively over time and you people are hungry, but we'll do 2 quick questions, then you can eat. If you have a question, please just raise your hand and someone will bring a mic. Any questions? Am I missing someone? Oh, sorry, over there.
Thank you, everyone. This is Nicola Holden from SRUC in the UK. I really enjoyed that because it's an area that's really close to my heart in agri-food production. So I would love to get any thoughts you've got about how we could potentially expand some of the templates that we've got here to that sort of true One Health context, you know, as we've heard, for livestock production or for crop production around biosecurity as well, where we need to protect the sensitivities of some of those stakeholders that we've just heard about. So what are your sort of reflections on that sort of aspect of expanding the public health out towards other One Health stakeholders?
Who would like to take that one?
Yeah, I guess I can. I mean, that's in a way what we're trying to do with the AusTracker platform, that different sectors and different stakeholders would see the data partitioned in different ways, which would allow those sensitivities to be considered in terms of not everyone would have access to that sensitive data, but there would be a degree within it's being placed in a context that is still meaningful, if that makes sense.
Okay, one more question. We have, yes, Emma.
So thank you to all of the speakers for really amazing talks in general. I wanted to maybe just drill into one of the points that I think many of you raised, which is basically, I'm going to quote Mira Chand here, if we don't know what's normal, we don't know what's abnormal. But I feel like even though so many of us know this and say this, it is very difficult to get policymakers to fund this or even support it, especially because though we talk a lot about One Health, in policy it's often still very siloed. You know, animal diseases are over there, human diseases are over here, and we just pretend like they might not ever interact. How can we convey this message better, and how can we get more support for the fact that we need better comprehensive surveillance? I know that's not an easy question.
Yes, that is probably the biggest question we have. Thank you.
I guess maybe if I can.
Yes.
So I think it's really just about that kind of communication, and you know we've been talking a lot about kind of giving credit where credit's due, and I think the thing that, you know, from a kind of human health perspective, which, you know, makes headlines and is really important to policymakers, is really on us to show the importance of the surveillance that happens outside of the human health space and how we're able to use that information to change our approach to public health. And if we don't do that and we don't make it obvious that without their contribution, none of this would have happened, then it's on us that we don't have the data we need. And I think we, you know, need to work with our counterparts to really amplify their value. And if we don't do that well, then we're going to lose that benefit.
Idrissa.
Thank you so much. I just would like to add one aspect that is important. But as we notice, most of the viruses of potential pandemic are from the animals. This means if whatever the effort that we are doing for surveilling human and others, if we are not including the part of the animals, it will serve nothing for us, because we will lose our time, we will lose our energy. For example, just our example, we are a good surveillance program in Senegal for human. Whatever the number of cases, we have the possibility to catch it up at real time. But if we ignore, for example, that beside the urban circulation of dengue, they have one part that is related to the salvific cycle, and then add a layer of complexity in our effort to mitigate this, we are wasting our time, and maybe the effort that we are doing will serve for nothing. And then we need to move towards the One Health approach and be able to survey at the same time the human, the animal, and integrate all this data for maybe forecasting or having a reliable effort to balance and countermeasures for these pathogens. Yes, thank you.
Yeah, no, absolutely. I completely agree there. Nicki, do you have anything you'd like to add? Okay. Everyone, there's lovely food outside. We thank you for your concentration, and again, a round of applause for this wonderful panel.